He called it the sleep hangover. Every morning, Gerald would wake up, make his coffee, sit in the kitchen, and for the first hour, feel like he was moving through fog. Not tired exactly. More like something hadn't finished loading. His wife described it as him coming online slowly. It had been that way for about 2 years. Gerald was 71. Retired engineer. Sharp man. Precise in his thinking. He had attributed the morning's slowness to age. His doctor, in a routine visit, had never raised it, and Gerald himself had never connected it to the small white pill he took every night at 10 o'clock to fall asleep. He'd been taking Zolpidem for 4 years. He didn't think of it as a serious medication. It had been presented to him as short-term help with insomnia after a stressful period at work. Short-term became a year. A year became 4. The prescription renewed automatically at each appointment, and nobody had reviewed it. I want to tell you what his neurologist found when she finally asked the question everyone had missed. But first, I need to show you a study published in January 2025 in the journal Cell, one of the most prestigious scientific journals in the world, that changed how researchers think about what sleep medications are doing to the aging brain. Not through the mechanism we discussed in the first video of this series, not through acetylcholine, not through anticholinergic burden, through something else entirely. Something that was only recently understood well enough to measure. Stay with me. Because what this research reveals is one of the most important findings in sleep science in the last decade, and it has direct implications for anyone over 60 who takes a sleep aid regularly. Let me start with the brain's cleaning system, because most people have no idea it exists. Your brain accumulates waste during the day, not metaphorical waste, literal molecular waste. Metabolic byproducts, misfolded proteins, cellular debris. Among these proteins are 2 that researchers have been tracking in Alzheimer's disease for decades, amyloid beta and tau. They accumulate during waking hours as a normal consequence of neural activity. The question that puzzled neuroscientists for years was, where does it go? The brain has no lymphatic vessels, the network the rest of the body uses to drain waste. So how does the brain clean itself? The answer came from Dr. Maiken Nedergaard's laboratory at the University of Rochester in 2013. They discovered what they named the glymphatic system, a network of channels surrounding blood vessels in the brain, through which cerebrospinal fluid flows, flushing metabolic waste out of neural tissue and into the body's drainage system. The glymphatic system runs primarily during sleep, during non-REM sleep, and specifically during the slow wave. Deep sleep stages, cerebrospinal fluid pulses through the brain at rates that are 10 times higher than during wakefulness. The brain is literally washing itself while you sleep. Dr. Nedergaard described it this way: it's like turning on the dishwasher before you go to bed and waking up with a clean brain. And the proteins being cleared include amyloid beta and tau, the same proteins that, when they accumulate, are hallmarks of Alzheimer's disease. Gerald, if he had known this, would have wanted to understand the pump. Because there is a pump. And that is what the 2025 study found. A research team at the University of Copenhagen, working with the Nedergaard Laboratory at the University of Rochester, wanted to understand exactly what drives glymphatic clearance during sleep, what the actual mechanical driver is. What they found was a specific mechanism that no one had measured at this level of detail before. During non-REM sleep, neurons in a region of the brainstem called the locus coeruleus release norepinephrine, a signaling molecule, in slow rhythmic waves. These norepinephrine waves cause the brain's blood vessels to contract and expand in synchrony. That synchronized vasomotion, the rhythmic pumping of the vessel walls, is what drives cerebrospinal fluid through the glymphatic channels. Without the norepinephrine waves, the pump doesn't work properly. Glymphatic flow slows. The brain's clearing process is impaired. Then they asked the question that changes everything. What happens to this mechanism when a common sleep aid is given? They tested Zolpidem, sold under the brand name Ambien, one of the most widely prescribed sleep medications in adults over 60. The results were striking. In mice, and I want to be clear that this was an animal study, which means the findings need to be interpreted carefully before applying them to human Zolpidem effectively induced sleep. The animals fell asleep more quickly. But the norepinephrine waves were suppressed by approximately 50% compared to natural sleep, and cerebrospinal fluid transport into the brain dropped by more than 30%. The sleep looked right on the surface. The cleaning wasn't happening underneath. The authors wrote in their paper that Zolpidem interferes with normal sleep architecture and suppresses glymphatic flow in mice, and that sleep aids like Zolpidem are unlikely to improve glymphatic function. This is animal research. The mechanisms in humans may differ. We do not yet have a human imaging study demonstrating this specific effect, but the mechanism norepinephrine-driven vasomotion as the driver of glymphatic clearance is well established. And Zolpidem's effect on norepinephrine dynamics is not new. It was the magnitude of the suppression, now measured for the first time, that alarmed researchers. The paper was published in Cell on January 8, 2025. It was funded by the National Institutes of Health, the Novo Nordisk Foundation, and the Cure Alzheimer's Fund. Now, is this the only evidence pointing in this direction? No. There is a separate body of human observational data. A population-based case control study published in the journal Medicine examining nearly 100,000 patients found that after controlling for age, sex, and a comprehensive list of comorbidities and medications, Zolpidem use was associated with a 33% higher adjusted odds of dementia. The study also found dose-response effects. Higher cumulative Zolpidem exposure was associated with higher dementia risk across most dementia types. Observational data cannot prove causation. People who take sleep aids chronically may have underlying conditions that independently raise dementia risk. The researchers acknowledge this. But the dose-response relationship, more drug, more risk, is the kind of signal that strengthens an association beyond coincidence. And it is consistent with the mechanistic picture the 2025 Cell study provides. Two bodies of evidence, one in animals, showing the mechanism, one in humans, showing the association. Neither is complete alone. Together, they tell a story that anyone taking a sleep medication nightly, especially after 60, when the glymphatic system is already less efficient, deserves to hear. Gerald's neurologist, when she reviewed his medication list at that routine visit, asked him one question. How long had he been on Zolpidem? When he said 4 years, she paused. When she asked whether anyone had discussed alternatives, he said no. She said that needed to change, but carefully. Stopping a sleep medication after 4 years of nightly use requires a gradual taper under medical supervision. The brain adapts and must be allowed to readjust. Gerald's taper took 8 weeks. During those 8 weeks, his neurologist introduced him to CBT-I cognitive behavioral therapy for insomnia. The American College of Physicians designates it as first-line treatment for chronic insomnia, above medication, not as a last resort. As the primary recommended approach, with clinical trials showing more durable improvements than medication because it addresses the underlying patterns rather than sedating them. Gerald was skeptical. He was an engineer. He wanted a mechanism, not a behavioral protocol. His therapist gave him one. She explained that chronic insomnia creates a conditioned arousal response, the bed becomes associated with wakefulness, and the brain prepares to be awake the moment it enters the sleep environment. CBT-I interrupts that association and rebuilds a new one. He understood it immediately. It's a firmware problem, he told his wife, and CBT-I reinstalls the firmware. Gerald began sleeping without medication 3 months after completing the taper. Not perfectly, not every night, but consistently. The morning fog, the thing he'd been calling the sleep hangover for 2 years, had lifted. He was coming online within 20 minutes of waking instead of 90. His wife noticed before he did. He told his neurologist, "I thought I was sleeping. I had no idea I wasn't cleaning." Here is the question to bring to your doctor if you take a sleep medication regularly. I'd like to discuss how long I've been on this sleep medication and whether CBT-I or a lower-risk alternative has been considered. I've read research suggesting that some sleep aids may interfere with the brain's waste clearance process during deep sleep, and I'd like to understand whether that's relevant to my current prescription. That question asks for a duration review, names a specific evidence-based alternative, and references a mechanism any physician following the current research will recognize. If your doctor hasn't seen the January 2025 Cell study, share the title. Norepinephrine-mediated slow vasomotion drives glymphatic clearance during sleep, by Hoagland and colleagues. That gives them the exact reference. In the next video, I want to shift from what's harming the aging brain to something that consistently protects it, at least in the people who tend to live the longest. There is a specific biological signature found in the gut microbiomes of people who reach 100 years old. It appears across Japan, Sardinia, and populations that have no other obvious connection to each other. Researchers are beginning to understand what it produces, why most people over 60 have lost most of it, and whether it can be recovered. Subscribe so you don't miss it. And if you or someone you love takes a sleep aid nightly and has never had a conversation about what it might be doing beyond putting you to sleep, share this video with them. Not to frighten anyone away from medication, to make sure the full question was asked. I'm Dr. Ben. I'll see you in the next one. The information in this video is for educational purposes only and does not constitute medical advice. Always consult your doctor or a qualified healthcare professional before making any changes to your medications, including sleep aids. Never stop or taper a sleep medication without medical supervision.